Addition of the tap motif to H-Gus might enhance delivery during DRT. We put the 11 amino acid TAT tag at the C-terminus of B-glucuronidase with a glycine spacer, which we called GUS-TAT. We looked at the uptake by MPS7 fibroblasts of GUS and GUS-TAT. Here you can see that wild-type GUS is taken up by the cells and completely inhibited by mannose-6-phosphate. However, the GUS-TAT is taken up not quite as well, but is not completely inhibited by mannose-6-phosphate. It's this portion of the non-MAN6B inhibitable uptake that we're interested in here. We next looked at the retention of enzyme in fibroblasts following uptake. So both GUS and GUS-TAT plus MAN6P were taken up by the cells and stayed inside the cell after the uptake for essentially the same amount of time. Is the uptake of GUS-TAT dependent on binding to cell surface proteoglycans? We used two known inhibitors of binding to proteoglycans, heparin and polylysine, and found that with increasing concentrations of each of these compounds, the uptake mediated by the TAT was inhibited. However, there was no effect on the normal wild-type GUS. Then we looked at the tissue distribution of GUS versus GUS-TAT 24 hours after infusion into the MPS7 mice. In some tissues here on the left, like liver, spleen, bone marrow, there was not really any advantage in using the GUS-TAT. But in a couple of tissues, such as the heart and the kidney, we saw an increased delivery of GUS-TAT in those tissues. This is a look at enhanced delivery and clearance of storage in MPS7 mouse kidney after 3 weekly infusions of GUS versus GUS-TAT. We looked at images from non-infused, GUS-infused, and GUS-TAT-infused animals. The top panels are naphthol ASB staining of beta-glucuronidase. So red color actually means beta-glucuronidase activity. In the non-infused animals, we see none of that red color. In the GUS-infused animals, we see a slight blush of color. But in the GUS-TAT animals, we saw a substantial increase in the amount of beta-gluc activity delivered in those tissues. Now looking at clearance of storage in the same tissue, we see once again the storage in the kidney. Treatment with GUS alone did not reduce the storage very much, but treatment with GUS-TAT almost completely reverses storage. So there was an advantage to using the GUS-TAT over the wild-type enzyme. So the conclusions from this is that GUS-TAT is taken up by fibroblasts in a MAN6B independent manner. The binding to cell surface proteoglycans such as heparin sulfate mediate the uptake by absorptive endocytosis. And delivery of GUS-TAT and clearance of lysosomal storage is enhanced in some tissues, especially kidney. The TAT delivery system, although it is slower, could also be used to deliver enzymes for bioremediation. Next, I'm going to talk about our use of an FC tag GUS for transplacental delivery to the fetus, which we published last year. It's well known that maternal IgG is transferred from maternal circulation across the placental to the fetal circulation. And this transport is by the binding of the FC portion of maternal IgG to the neonatal FC receptor, or the FcRn. This binds to the FcRn and is transcytosed across and released into the fetal circulation where it has immunological function. A similar system works in the intestinal cell, the neonate, where IgG in the mother's milk is transferred by the same process, utilizing the FcRn, delivering it across to the newborn. So we thought that it might be advantageous to use or possible to use the FC domain to deliver human beta-glucuronidase across the placenta. So what we did was put the CH2, CH3 domains of human FC onto the C-terminus of beta-gluc, once again separated by glycine spacer, which we call GUS-FC. First, we wanted to see if GUS-FC had a functional FC domain. Here we did a simple assay using protein G sepharose, which protein G, of course, binds to the FC domain and would be able to precipitate it out of solution. If we put GUS into that assay, very little of it is precipitated by the protein G sepharose. However, GUS-FC from the medium and after purification, a substantial portion of those molecules contain a functional precipitable FC domain. We next looked at the plasma GUS activity in newborn MPS 7 pups from pregnant females infused on embryonic days 17 and 18 with GUS, GUS-FC, or a long circulating form of GUS called PERT-GUS, which I'll talk about later. Here we looked at the total number of moms and the total number of pups. And here in the last column is the plasma GUS in pups on newborn day 1 in units per mil. Here we can see that in mothers that were infused with buffer only, the pups had the low level of B-glucuronidase seen in the deficient state. If we infuse the mothers with GUS, we also saw the same low levels of enzyme. However, in mothers that were infused with GUS-FC, the pups now contain a very high level of enzyme, indicating that that enzyme has been transferred across the placenta and delivered to the pup prenatally. And lastly, the long circulating PERT-GUS, which is in the circulation for the mother for a very long time, that was essentially not transferred across the placenta. So we wanted to confirm that the transplacental transfer is FC domain dependent. So we reasoned that GUS-FC should not cross the placenta of pregnant females that are FcRn knockout. And since the FcRn is the MHC class 1 protein, beta-2 microglobulin, which is an accessory protein for all of that class of proteins, if the moms were beta-2 microglobulin knockouts, they should be functionally FcRn knockouts. So they also should not transport GUS-FC across the placenta. And we also used a mutant GUS-FC that contains 3 mutations in the FC domain that are known to mediate the binding of FC to the FcRn. We next looked at transplacental transfer in the FcRn knockout, the beta-2M knockout, or normal mothers infused with GUS-FC triple mutation. And these animals are not MPS 7, so they're not beta-glucuronidase deficient. So in GUS-FC infused into a cross mothers or cross a B6 by B6, the pups have an endogenous level of enzyme that's about 114 units per mil. So transplacental transfer would be anything over and above that level. So when we infused enzyme into mothers from a B6 by B6 cross, the pups once again had transport of enzyme across the placenta. If we looked at either FcRn by FcRn or beta-2M by beta-2M crosses, the pups from those crosses essentially had no additional enzyme, so there was no transport. And interestingly enough, if we cross the FcRn knockout with normal B6 and produce pups that had half the normal levels of FcRn, they had about half the transport. And lastly, in the triple mutant GUS-FC into B6 by B6 cross, where the animals have normal FcRn levels, there was no transport of that mutant enzyme. We next looked at the GUS levels in perfused tissues from newborn pups from mothers infused with GUS-FC on embryonic days 17 and 18. So we looked at these tissues. These are the wild-type levels in non-infused wild-type B6 pups, just for comparison. But in the pups that were from mothers that were PBS infused, we see the low levels of enzyme. Now these are MPS 7 animals again, so they're deficient in beta-glucuronidase, so they're at low levels. If we infuse GUS into the pregnant mothers, we see essentially no increase. However, in the mothers that were infused with GUS-FC, we saw substantially delivery of this enzyme to the various tissues.